is just your announcement.
This is my boss, Steve.
So is this the family?
This is the band.
I'm the Sam.
You're the Sam?
I'm not Sam.
Oh, okay.
John's from church.
Okay.
Sam is Sam.
Uh-huh.
Nick is James's dad. This is like a test.
Okay.
Everybody freaked out. This is my papa. You can call him Gary.
Oh, hi.
Probably might call him Papa.
Tends to talk afterwards.
Yeah.
Various things.
Okay. Do you know how to lock this door? I'm afraid that if I close it, people get locked out. Well, I've seen Deb do it, so I don't know if you can just do it with you. Hey, Mom.
Yeah, listen in while I'm listening in.
No, I'm just gonna make sure it's working, then I'll put it away.
Well, I don't leave it open because it makes people go down the hall with the information. But if it's locked, it can go to
I've never seen anybody lock it where
How many scientists?
Duct tape.
Maybe it's not doable. James wants to just duct tape it. We can also just leave it alone. That's an option. I'm speaking.
Always just leave it right.
Well, I hate when this door is locked because sometimes, like, I need to go to the restroom and come back in and the door is locked. A personal pet peeve about.
Put it where you would like.
We are live on YouTube. Can you hear me, YouTube?
Hey, Mo.
How are you?
A little frantic. Everything I own is in yellow boxes.
I was gonna say, if you're in good mood or.
Yeah, the movers arrive at 8 a.m. tomorrow morning.
Nice.
And then after that, I saw the
I own a lot of junk.
Maybe this is a good time to clean up a little.
I have thrown away 10 large dumpsters
Oh, really?
of stuff.
You're live on YouTube.
Ooh, hey, YouTube. Your screen is black.
Well, I don't want you to get.
And then,
Hi. You're so much more excited than me. Susie, yeah.
You have time to be excited too.
Yay, I hope. Hopefully. It's the hope.
Hi. I'm so glad that you want to go to that playing thing. I saw it on your bar. I was like, oh my god.
Are you going?
Yeah.
I'm excited. But we're not going to like
I was like,
January? Oh, that's
But
cool.
yeah, no, I was just thinking, I was like, man, I need something green for my desk. It's like kind of boring.
It's my fault. I wanted a particular, they came in like boxes for the glass spheres, and I wanted the spheres.
This is the dreaded like 10 minutes before your talk where you can do nothing. You're just sit there.
Just sit there and wait. Well, you had such good company too.
That's right.
I do.
All the bright smiley faces.
Yeah.
Yeah, I think I'm only bright and smiling because I'm on the other side of this.
How long ago was your defense? 2 years now?
It was like, no, it was like a year and a half.
Because it was June?
Yeah, it was like July, early July.
Hey! This is Audrey. Have you met Audrey?
I am not.
I've spoken about you.
Nice to meet
Good to meet you.
Yeah, I was just thinking on my way here, like, that's like an hour of my life that I do not remember a single thing.
You just blocked it out?
Yeah.
That's what I've heard.
I'm moving tomorrow.
Moving. That's so very exciting.
This will be exciting once it happens.
Hey, George.
Wow. So it's all packed up.
Pretty much.
Hey.
I mean, all the shelves are in.
That's
Oh.
pretty.
What?
We moved all,
Can't
we moved
really-
all the
We have beer.
I've had a beer.
No, that's fine.
microscopes.
It's the glasses.
It's the glass.
It's the glasses.
Normally I'm like
Normally
this.
it's-
Oh, okay.
Yeah.
More than this. And I was walking
My
over there, I
mom is
walked
so
down.
mysterious. So that's why we were gonna stream anyway.
I thought it would be a stream.
Oh, okay.
George, are you working the week after?
Yes,
Oh, okay.
This is my eye.
We're taking us. Yeah, I'm gonna come hang out.
Yeah. Very, very, very many. These
So are they going back to
I think it would be nice.
How long?
Actually, it's all open.
That
lab.
was the
All excited. Whoa.
There's a, the cake thing for December, was it 1:30?
I saw you guys.
I know that somebody had a video, I also
Well, I
watched
didn't.
it. Sent me a note.
Yes, it's already on.
I was able to make the live one.
So we're training from the computer.
Watch the cake.
I saw, I mean, I think goes to other things. She's like a, kind of like a white lab. I hate we have one whole lab. I will be like, but she looks like her face looks like a lab.
In general.
I'm not even, why don't you like, I think it just tastes like sugar. It tastes like it's like,
Hey,
it's not, it's like cheese. It's gonna be I just hate ice cream, like, a lot.
It grows, it's too close.
It's good.
I got a lot of pictures in my time. I got Karen.
Right, right.
Did you not lock that door? She's unlock the door. Well, let's see you do it.
You get to,
Teach me your
I would invite you if I would stay, but next time you're.
No, it's fine.
Yeah, we like hold it.
I'm free. I can do whatever I want. I can go
When
anywhere.
I travel, I found, she thought about it for a while.
Then go with the, I think that was the price of those.
Have a bunch of Chinese UN. So that was interesting. So it sounds very interesting.
It's red and, but once you figure that out.
Sounds almost all my old friends.
Window breaks.
Oh,
It must.
so I'm happy to say,
Yeah.
but it's
But yeah, it's right.
I mean, the very first one.
And I don't know.
Yeah.
When I am not,
Yes.
And then
I was a new faculty member. I hadn't even gotten the whole months or something. Chair of the partnerships. Right.
Yeah, I got to.
And you go send it to me and I'll read over it. See what better. So that's like all the Christmas big there. I'm done.
Hey, Sam.
I just slow it. 3 months. I got it.
That's good.
Yeah, I found first.
Hey, Jason. Thank you for your comments.
Oh,
Oh, no, I was also talking about the.
Oh, yeah, how did that go?
Went well.
Easy.
I have long emails. I just go.
yeah, I was gonna say.
I can pack this one. I mean, I like.
My mom.
My proposal for non-selective is like to. Can you explain to me what in this house?
We are kind of like this, long. The seminar.
So I got several emails, I mean, I got somebody from Denfense that said, you know, I knew I really loved to talk. That was the whole thing. So, I mean,
Okay, so it's the anointed hour. It's my pleasure to take the prerogative of the thesis mentor to say a few words before Tiffany's seminar. Tiffany's been a terrific student in the lab, done some great work. And so it's always a challenge of what do you say about somebody like this? And so the first thing I'll say about Tiffany is I don't think she's ever boring. It's very exciting. The lab has just got a different sort of a sense when she's around, like in many kind of ways. There's a lot of energy. There's a lot of odd things that happen in my lab that some of you may not be aware of, one of which is the Lab Olympics, and she was competing in the Lab Olympics. As you can see on the right, she didn't get the gold medal, but she was.
Marcus cheated.
There you go. There you go. I could see something about competitive spirit. There you go. Things like that. So the other thing that's that you get into in my lab is odd sort of things like this. And so this was an example of Tiffany mastering the flamingo position, being ably guided by a former student, Britta Henderson, in this regard, and things like that. And as you can see, she's developing all the criteria for being a true member of my lab. The other thing about Tiffany is that she brought a sense of style to the lab that you not oftentimes see in many graduate students and things like that. She's always a little more dressed up, and she actually is starting to take it to the next level in terms of bringing it into the high culture mode of these things. And nothing would be complete with Tiffany without shoes, and of course this is her matching line of shoes to go with these sorts of things like that. These are by far and away not the scariest shoes we've seen in my lab, but we won't go into this. So what else can I sort of say about this? So one thing about Tiffany, she's got tremendous drive and persistence. I think as some of you know, she had some health issues that caused her to leave the program. She didn't give up. She came back and started anew and did terrific work, which she'll tell you about it. She has a lot of drive and persistence, and I think a lot of the credit for that goes to her. And the last thing is she really is a scientist of substance. She took on a very nice project in the lab, something that we've never worked on, which is flagellum biology. You always have to have a special student to go off into something that your advisor knows very little about, even though they may think they do, and things like that. And she really developed some nice a story that she'll tell you about. And one of the benefits of this was that we had made, I always we or I, I'm not sure which, had made several predictions about the phenotypes of these mutants. And as is typical when we make predictions about Leishmania, both of these predictions were wrong, but the nice thing was, in fact, the outcome of these things turned out to be more interesting than our original predictions, so we were all pretty happy. So with that, I'll turn it over to Tiffany, who will tell you about her story with the Leishmania flagellum.
Thank you.
Gotta get that dress. Thank you, Steve, for the introduction. That exceeded my expectations. And all my colleagues who made the time to be here today, and my family and friends in attendance. And some of my family is not here, but we have a live stream going, thanks to my husband. Hello, YouTube. But before I get started on my thesis research, I want to spend a few minutes defending to the non-parasitologists in the audience why I chose to spend the last 3 years of my life working with parasites. I bring this up because when I first got to graduate school, I was in a breast cancer research lab. And when you do breast cancer research, the unanimous response when you tell people what you do is something like this. But then when I had to tell people I did parasite research, I got very different reactions from most people. This actually bothered me a little bit because I think part of our job as scientists is to be able to explain the beauty and the relevance of our work. So I spent a lot of time mulling over why I got this disgust sort of reaction from people. And I've concluded it's because when people hear parasite, they envision something like this. What I'm envisioning is actually something like this. So here are dozens of Leishmania parasites inside of a single macrophage cell. And macrophages are the cells that are part of our immune system, and their job is to go through our body, identify and kill potentially harmful pathogens. But Leishmania choose to live there. They thrive there, and it makes it its home. Another interesting thing is the size. Leishmania is single cell. It's approximately 2 to 3 micrometers. To give you a sense of what that means, this bar represents 10 micrometers, which is the same as 1/10 of a millimeter. So they're strong but mighty. To put that into context, the width of a human hair is 70 micrometers. So that's like 30 Leishmania parasites end to end. Or if you use the example of malaria, since everyone knows malaria, that's approximately 70 malarial parasites. So while human beings evolved in these multicellular organisms with these complex neuronal networks, capable of the most advanced technology, parasites evolved as well. These are all examples of single-cell parasites that are human pathogens. So they infect us, they humble us, and they're actually among the world's most successful and sophisticated organisms, which that's how I feel about them. So getting on to the research. As Steve said, I work on the flagellum, and I work on the role of this organelle throughout the Leishmania life cycle. To start with a primer, Leishmania causes leishmaniasis, which is a neglected tropical disease as defined by the World Health Organization. There's over 20 different species of Leishmania that can infect us, and depending which species infects you, you get different symptoms, which we can categorize into roughly 3 groups. Cutaneous leishmaniasis is where the site of infection, you can get an ulcerated wound. Mucocutaneous parasites metastasize to the nasopharyngeal tract, and the inflammation causes disfigurement, as shown here. And then visceral leishmaniasis, a species causing that is Donovani, which I use for my work. Visceral leishmaniasis is when parasites metastasize to the internal organs, primarily the liver and spleen, and it can be fatal if left untreated. It causes approximately 20 to 40,000 deaths annually, which is a mortality rate of like 10%. And this mortality rate can experience booms in time of famine or in times of war. An example of this being in Sudan last year when civil war broke out. We saw an increase in deaths from this. Although most Americans have not heard of leishmaniasis, it is pretty pervasive. Endemic transmission occurs in 98 countries, grouped mostly in South America, the Middle East, and countries in the northern half of Africa. Something interesting I'd like to point out is there's a large disparity between the number of people that are reported infected and the number of people at risk. And there's a few reasons for that. One, this is a neglected tropical disease, so it occurs in 4 countries. Also, there are a large number of cases that are asymptomatic. So when you think about it from the parasite's perspective, they don't want to kill their host, so it's pretty well adapted for its job. The life cycle is digenetic. So there are 2 developmental stages. There's the promastogote shown here in these GFP-expressing parasites, and those are found in the insect vector. So the vector goes from host to host, carrying the parasite. When the sandfly feeds on a vertebrate host, it regurgitates parasites into the bloodstream, which are then taken up by macrophages. It is within the phagolysosome that differentiation occurs to the amastigote stage, shown here. And of course, the cycle continues when a sandfly feeds on an infected host. It's a little more nuanced than this. I'm going to give you a bit more detail that I think is relevant for the data I'll be presenting to you. There are different morphologies of promastigotes. There's two that we can culture in the lab, so we mostly talk about those. There's procyclics and metacyclics. Procyclics are found in the gut of the fly. They can replicate, but they're not infected. Metacyclics are found in the mouthparts awaiting transmission, and they are infective, but they cannot replicate. It is these metacyclics that are picked up by the macrophages and differentiate to the amastigotes, which are not only infective, but also can replicate. So this causes the brunt of the disease in humans. Here's an image shown to scale. I'm showing you this so that you can see the morphological differences between promastigotes and amastigotes. It's very striking. The promastigotes are larger. They have an elongated cell body. In contrast, amastigotes are much smaller and rounder. But the most important thing is this single flagellum. Promastigotes have a relatively long flagellum and aids in motility. They swim flagellum first. But in amastigotes, there is significant reduction in the length of this organelle. For a long time, amastigotes were misnomered as a flagellate. I even still see this in the literature on occasion, which is irritating for me. But a flagellum is clearly present. So, like I said, I focus on the flagellum. And flagella and cilia are found ubiquitous organelles in eukaryotes. As a side note, flagella and cilia are the same, but those terms were applied before the structures were studied, so if I use those interchangeably, that's why. Flagella protrude from the cell surface into the extracellular environment, and because of that, they're often sensory organelles, and they can be motile or non-motile. I'm showing you an overview of the diversity of flagella. So left to right, we have the epithelia of the ventricles of the brain. We have a mouse kidney collecting duct cell. There's a single-celled paramecium covered in cilia. There's the single-celled organism Trypanosoma brucei, and then Leishmania. So from this global view, you might be led to believe that the structure of flagella are variable, but actually the core structure of flagella are conserved. And I'm showing you that here on the left. So if we break the flagella down to its most basic parts, within the cell body is the basal body. It is from the basal body that the microtubules are nucleated that form the axoneme. The axoneme is made of stable microtubules. It runs along the entire length of the flagellum and is its core, and it also has a flagellum membrane. If we take a slice through the middle of an axoneme and look at a cross section, I'd like to point out some of the microtubule arrangements to you. So there's two overall axoneme architectures. There's more, but the vast majority fall into one of these two categories. Motile flagella have a 9+2 microtubule arrangement. The 9 refers to these 9 outer doublets, each having an A and B tubule. The +2 refers to the central pair. And it's the central pair plus accessory structures like outer diamine arms, radial spoke proteins, and inner diamine arms that allow for motility. In contrast, primary cilia lack this central doublet and typically are non-motile. But regardless of which axoneme type is formed, all axonemes require interflagellar transport or IFT for assembly. IFT was first discovered in Chlamydomonas, which is a model organism for flagellar biology. And that was in 1993. In this transmission electron microscopies, they saw electron-dense trains between the axoneme and flagellar membrane. And we now appreciate that IFT is essential for the assembly of almost all flagella, regardless of if it's a motile, non-motile, 9+2, or 9+0. How it works is IFT forms trains, if you will, at the base. These stable complexes are moved by motor proteins out to the growing tip of the flagellum. So they transport cargo like axonemal subunits, or they transport receptors that are flagellum membrane localized. Conversely, retrograde transport returns signaling molecules back from those receptors or accidental subunit turnover products to regulate the length. Another important component is the BB-zone. So BB-zone is typically not required for assembly, but it is needed to get the right stuff into the flagellum, particularly regarding trafficking of specific flagellar cargo for membrane localized receptors. And it's important to localize these membrane proteins as they function in sensing and signal transduction. Remember, the flagellum is in the extracellular environment. And mutations in the BB-zone and IFT can prohibit their localization and therefore their signaling function. These mutations become particularly problematic in primary cilia, causing ciliopathies. Ciliopathies have their consequences because of defective signaling in primary cilia. Primary cilia are typically not motile, like I said, but they are important as antennae. So they sense extracellular signals and then activate downstream signaling pathway cascades. And the primary cilium has crucial roles in vertebrate development. So if the primary cilium is messed up, we have developmental disorders with pleiotropic symptoms. So this can lead to cognitive development, polydactyly, situs inversus. The most commonly known one is polycystic kidney disease, for example. And the reason that I keep talking about axoneme structure is because, as shown here, the structure correlates to function. And in the Leishmania parasite, there are different axoneme structures between the promastigote and the amastigote. Promastigotes are motile, like I said, and their axoneme reflects this. They have the 9+2 axoneme. Also alongside it, you'll notice this unique structure you've probably never seen before, and that's called the paraflagellar rod. It runs along the length of the axoneme, and we know that it's required for proper motility in the parasite. In contrast, the amastigote, which is non-motile, lacks the +2, so it's only got those 9 microtubule doublets. It also lacks the paraflagellar rod and those accessory dynein arms and other structures needed for motility. So maybe these different axonemes can give us a clue as to the function of promastigotes and amastigotes. There are some structures maintained throughout the life cycle, however, and if we zoom in on the region where the flagellum exits the cell body, I can point those out for you. So what you're looking at is a slice through the length of the cell body. Here's the flagellum. This is the axoneme portion with the basal body. The basal body is physically connected to this organelle called the kinetoplast, and it houses all the mitochondrial DNA for the cell. The flagellum extends out through a flagellar pocket, which is approximately 2 micrometer deep invagination of the plasma membrane, so it forms its own distinct subdomain. There's 2 things I really want you to take home from this slide. One is the flagellar pocket structure. Because its structure may have important implications in its function, the flagellar pocket is the sole site of endo and exocytosis for the cell. So the rest of the cell body is corseted with a dense network of subfollicular microtubules, so it all happens through here. Also, the flagellum is kept throughout the cell cycle. It's not broken down and then rebuilt after cytokinesis. It's kept throughout. Because of that, it's been presumed that the flagellum assembly is essential for proper kinetoplast segregation during cell division, and therefore it may be essential for viability. That the flagellum needs to be assembled to maintain that flagellar pocket structure and also its function, consequently. There's also a correlation between flagellum length and cell size. And if there are flagellum localized receptors, they may be needed for downstream cell signaling throughout the life cycle. There are proposed stage-specific roles as well. We know in promastigotes the flagellum aids in motility. That one's straightforward. It may also have a role for attachment to the sandfly gut. There were these elegant EM studies done in the 1970s where they showed the flagellum between the microvilli lining the epithelia of the sandfly gut. But there's no functional evidence for that. The only other evidence to support that was work done in vitro, where they showed the tip of the flagellum can form these tight desmosomes with plastic, but again, not functional. It also may have a role in macrophage entry. Time-lapse microscopy showed macrophages take up the parasite flagellum first, but they swim flagellum first, so this is also not proven at this point. We know even less about the amastigote stage, in large part because it was little studied for a long period of time. In 2010, there is a hypothesis that was put out that the flagellum may play a role in virulence within the host, so it may actually be critical during this stage. And that was posited due to indirect evidence, basically. One piece of evidence being the axoneme architecture. Again, it's akin to those primary sensory cilia. It was also shown that amastigotes within the parasitophorous vacuole of the macrophage, the tip of their flagellum comes into physical contact with the host of that membrane. So the tip of the flagellum could provide a platform for host-parasite interactions via receptors, presumably. Also, remember the flagellar pocket is the sole site of endo and exocytosis. So it may be oriented toward the host for release of virulence factors through the pocket or even through the flagellum itself if it's acting as a secretory organ. So since there are a lot of unanswered questions but presumed roles, I asked if we could generate an aflagellate mutant to study these constitutive and stage-specific functions. So I needed to disrupt flagellum assembly. And to that end, I targeted the intraflagellar transport pathway. There are a few reasons why I did so. One is all of the IFT components are conserved in Leishmania. Another is the IFT components form these biochemically stable complexes. So if you get rid of one of those core components of the complex, you disrupt the entire complex and block the IFT. Also, disrupting IFT is typically not lethal, so the cell doesn't need it. An exception being Trypanosoma brucei, or T. brucei. T. brucei is a trypanosomatid. It's a fellow of Leishmania in that regard. And they found that flagellum assembly is essential for viability in that organism. They found that by doing inducible RNAi targeting different IFT components. And while T. brucei is a model organism in flagellar biology, Leishmania we know a lot less about. So a lot of the presumed roles in Leishmania are posited based off of the evidence found in T. brucei. But while there are similarities, there are obvious differences as well. One of those being the free flagellum. It's not attached to the cell body. So my first goal was to determine if I could even disrupt flagellum assembly and get live cells to work with. So for that, I inhibited flagellum assembly by disrupting IFT, as I said. Specifically, I chose a component, so I knocked out the retrograde component IFT-140, first in mastigotes. And then I used Leishmania donovani strain Bob, or LD Bob, named after someone named Bob. So LD Bob provides several advantages, the most important of which is I can readily culture both premastigote and mastigote stages. So I want to look at the flagellum throughout the life cycle, not just premastigotes, which is what is typically done in our field. We do this by emulating conditions that would be encountered in the macrophage, and by azenic, what I mean is cell-free. So there's no additional macrophages or anything needed for this culture. Other advantages include a sequenced genome. We not only have a sequenced genome, but I have sequenced the parent strain that I use for all the studies I'll be showing you. We have a mastigote stage-specific markers to validate that our azenic mastigotes are truly mastigote-like and not just round, angry cells. And for virulence assays, we have both in vitro and in vivo models. So I first attempted to generate the IFT-140 knockout using double allelic replacement by homologous recombination with drug resistance cassettes. And while I could get both of the replacements to occur, there was always a copy of IFT-140 around, likely due to induction of aneuploidy. But I was able to delete both copies in the presence of ectopic IFT-140. So here I have the chromosomal null background with the episome. This can be a clue that the gene is essential, and that happens in Leishmania, but it also has a plastic genome, so it could just be hard to knock out. We've encountered that before in the past too. So we have a test for gene essentiality using plasmid segregation. So how this works is you remove the drug selection against the plasmid, and then you test for episome loss using a marker. In my case, I used the GFP that's present. We were expecting the gene is essential, that it's needed for viability, so it would be retained after we remove the drug selection. So I removed the drug selection, and then I did my sorting with the GFP. On the right is the chromosomal null. On the left is a positive control. I use this because the heterozygote should readily lose the plasmid. But in both backgrounds, I got a large GFP DEM population. So these parasites either have very low expression or the episome is gone. To test if the episome is gone, we look at survival of clones plated out in 96 whole plates. And we got good survival, and the plasmid was indeed gone when I tested by PCR, shown here on the bottom. The point of all of this was to try and get aflagellate cells to study. And by these two imaging modalities, the flagellum is indeed gone. You see no flagellum assembly external to the cell body and the IFT140 knockout. Also, they don't swim, which would be expected. They settle out in the bottom of the flask. An important point here is that the add-back complements, so it functionally complements and the flagellum is now present. And this is just quantification of that. So for the add-back, the mean is not significantly different in length or in the distribution of lengths. This might confuse you a little bit. The reason it's heterogeneous cultures, so they're not all exactly the same length. They're at different points post-cell division, so they have different flagellum at points. However, a large portion of the flagellum is located in the flagellar pocket, so I cannot call these cells a flagellate without looking within the pocket. So to look in the pocket, I did transmission electron microscopy. And there's two views. There's the longitudinal view through the cell body, and then there's the cross sections. And depending on where it's cutting through this pocket, you'll see different microtubule arrangements. So the basal body has the 9 triplets. There's a transition region in the basal body that doesn't have triplets, but it doesn't have a central pair either. And then the axoneme or flagellum proper will have that 9+2 structure, because these are the promastigotes. In longitudinal sections, these cells are indeed a flagellate. I don't see any assembly of an axoneme, but it's important to note the basal body is still present. Also, the basal body is bald, if you will. There's not a swelling of material inside of there. And then by cross-section, I never observed that central doublet in the knockout. And the add-back did complement that. Basically, the conclusion here is that I have live cells, which was exciting. I expected them to be dead, but they were alive, so I could test a lot of those proposed roles using these promastigotes. So I tested if the flagellum is required for cell body morphology, if they not only are viable but also grow normally, and if the flagellum, and if the flagellar pocket structure and function is altered as well. So the cells are not only viable, but they have a normal doubling time in culture, shown on the left, and they're healthy, shown on the right, as determined by propydium iodide exclusion assay. At the stationary phase, there is some decrease. Also very interesting was the cell body morphology. This was exciting to see. So even though the flagellum was gone, the cells are not rounding up and becoming a mastigote-like. They maintain that elongated promastigote cell body shape. Another unexpected phenotype was the flagellar pocket is still being formed. You see here that it still has this bottleneck-like structure, this narrowing. And I wanted to further quantify this by looking at different regions of the pocket. So I looked at the width of the pocket region. I quantified the length of the entire flagellar pocket, the width of that neck region, which seems that it would be defined by the flagellum width, and then the collar region length as well, which I'll talk about in a second. So even though the flagellar pocket was still built, it was wider and shorter. But the neck region is the one region that did not significantly alter in its width, even though the flagellum was not there to define that space. So it seems that flagellum needs to be present for flagellar pocket elongation, but not shape definition per se. And a clue as to why it's not needed may be because of this collar region. So this electron dense region here. When I quantified, it was not significantly different. And what I'm showing you here are a few examples of that collar region in the knockout. I'm going on about this because in T. brucei, this electron dense region is termed the flagellar pocket collar. It's the point where the flagellum exits the flagellar pocket. And in T. brucei, assembly of the flagellar pocket collar is necessary for assembly of the flagellar pocket. So to confirm that this collar region is the Leishmania equivalent of that seen in trypanosomes, I performed endogenous tagging using YFP of the flagellar pocket collar localized protein bilbo-1. So nobody has actually assessed any of the flagellar pocket collar proteins at all in Leishmania. So this is immunofluorescence in the wild type background and also the knockout. I saw the signal came at the anterior region of the cell, exactly at the point where the flagellum exits. So this is the flagellar pocket region. Interestingly, the knockout also had the same localization and fluorescence intensity was the same. So maybe this is a clue, the flagellar pocket collar versus the flagellum is more necessary for building that flagellar pocket structure. Another thing we observed when we looked at this flagellar pocket was an accumulation of vesicles. In most of these EM images, I see accumulation of more than 2 vesicles. They're heterogeneous in size and color. Also in scanning electron microscopy, approximately 25% of the cells, there's accumulation at the anterior end outside the flagellar pocket, sometimes with these filamentous-like structures. Which begs the question, what is accumulating in the flagellar pocket? We know this is an important region for the cell. So I asked if flagellar pocket function is disrupted since it is the exclusive site of endo and exocytosis for the cell. There are different markers we can use to look at exocytosis. One is the glycocalyx. It is a complex surface that goes around the entire cell body and it comes out through the flagellar pocket. It's not poking out through the plasma membrane. It's a little deceptive. And there's also secreted acid phosphatase that we can look at too. When we looked at the glycocalyx by transmission electron microscopy and ruthenium red staining, I was able to quantify the width of this glycocalyx. So that's the really dark staining you're seeing here. There was no significant difference in the knockout. The major surface component of the glycocalyx is lipophosphogycans, or LPG. And LPGs contain these phosphoglycan repeats. And those phosphoglycan repeats are present in secreted acid phosphatase as well. So I used an anti-PG antibody to perform Western blot analysis to look within the cells at the SAP and LPG. And that's quantified here. So there was no significant difference in the abundance by densitometric analysis for the LPG region in the knockout or the SAP region by Western blot. There is this electrophoretic mobility shift though. And then I also looked at the conditioned medium. And to do that, I did a SAP activity. So this is in gel, native gel assay. And that's the only time that I saw a significant increase, is about 2-fold increase in the amount of SAP in the conditioned medium, which likely doesn't explain this massive accumulation effect that we're seeing. I also assayed endocytosis using a lipophilic marker, FM4-64. How this works is it's a pulse chase experiment. So 2 minutes post-chase, you see accumulation of the dye in the flagellar pocket. And the knockout was able to take up the dye. You see it in that flagellar pocket region, and it has the same fluorescence staining intensity. By 2 to 40 minutes post-chase, it enters the endolysosomal system. So this is the 2 minutes showing you wild type knockout and add back is mostly found, well, the majority is in the flagellar pocket. By 10 minutes, we're seeing signaling, or we're seeing fluorescence in the cell body as well as the flagellar pocket, so L is for the lysosomal system. And then by 40 minutes, the majority is located up like it should be. So in conclusion for this, endo and exocytosis still occur. And the next question was, what about metacyclogenesis? So if you remember, I told you we can isolate both procyclic and metacyclics in culture in the lab. So during in vitro culture, log phase parasites are the procyclic forms, but when the cultures are allowed to reach stationary phase, a portion of the population are these metacyclic forms. In addition to the change in morphology, there's also changes in the glycocalyx for the metacyclics. They have approximately 2-fold increase in the number of PG repeats. And these changes in LPG allow the metacyclics to be released from the sandfly gut so they can get to the mouth parts for transmission. Because remember, those would be effective forms. And importantly, it protects those metacyclics from complement killing once they reach the host serum. We can isolate the metacyclics from the procyclics in those stationary cultures using LPG-dependent or independent methods. I chose an independent method since we did not know if the metacyclogenesis was normal in the knockout. So I used density gradient centrifugation and was able to isolate metacyclic-like morphologies. This is shown here. So the knockout procyclics and the metacyclics are longer. I also assayed the LPG using that anti-PG antibody. And in the metacyclics, you see the increase in the size, as should be. Important is the functional assay. So I looked at the percent of viable cells when exposed to human serum. Here are the procyclics, and the metacyclics are the dashed lines. When exposed to 3% human serum, procyclics almost all died. However, the metacyclics were resistant, and the knockout was as resistant as wild type. So this was exciting because we could purify metacyclics, and we could then use those to study parasite infectivity. So we could actually do virulence assays with these aflagellate lines. So in summary, thus far, the cells were viable, so the flagellum was not required